Start again. The next speaker is Raymond Goldstein, who will explain how a Volvox embryo turns itself inside out. Thanks.
Thank you very much. And while it's usually the case that everyone says thank you very much to the organizers, I also want to not only thank the organizers, but also the steering committee of the Newton Institute for having the wisdom to sponsor this program, which I think is incredibly timely. So I've commuted quite a long distance this morning from the pavilion 100 meters away. And before I start, I should tell you about rule number 1 in Cambridge, which is that the names of departments and what goes on inside of them have nothing to do with each other most of the time. So this department, this mouthful of a department where I sit, is home to people like Stephen Hawking and people who do numerical analysis and people who work on ice physics and all sorts of things, including biological physics. And although it sounds very theoretical, I'm going to tell you today mostly about experiments that we've done in my lab, which is just 100 meters away. And I'm going to talk about a problem which is a bit of a mouthful itself, how a Volvox embryo turns itself inside out, but the subtitle perhaps gets the message clearer. This is really probably the simplest example of a topological transition in biology involving cell sheets. And it's the work primarily of two of my experimental postdocs, Stephanie Hone, who's a biologist, and who did her PhD in one of the leading biology groups that study this particular phenomenon, and Aurelia Honerkamp-Smith, who is a physical chemist by training and now an expert on imaging and membranes and all sorts of microscopic things. And two of my theory students, Pierre Haas, who's now in his 3rd year, and Philip Cook-Trong, who's spent most of his time doing other things for his PhD, which he just recently finished, but also participated in some of the analysis I'm going to tell you about. Now, before I begin on this particular problem, since we have an hour or so, I thought I should try to put this in context so you understand where we're coming from, why this problem of all other problems to study. And the best way is to tell you a little anecdote. And some of you might have heard me say this before, but it's just so much fun to recount, I can't resist. About 10 years ago, I was at a major North American university, which I will not name. And I was talking there with an astrophysicist. And you know how it is, you visit a place and people say, oh, what do you work on? And you say, oh, what do you work on? And I said, when he asked me what I worked on, I said, well, a certain amount of biological fluid mechanics. And he looked me straight in the eye and he said, is fluid mechanics important for biology? And I really didn't know where to begin. You know, I thought of maybe the heart or the lungs or the ears or whatever. So I thought a lot about how I should really respond to this somewhat dim astrophysicist. And then I realized that actually the answer was in Leonardo. So if you go back to this beautiful drawing, the Vitruvian Man, which Leonardo drew in the late 1400s. Of course, we've all seen this a million times, and for us, most of the time, what we see here are the wonderful proportions of the human body, the circumscribed and inscribed squares and circles that tell you that your wingspan is about the same as your height and things like that. But it's also a very powerful reminder of the bilateral symmetry that most of us have to a good approximation. And is common for most vertebrates. But as they say, symmetry is only skin deep. And in fact, if you look just beneath the skin, what you see is that all of us have a fundamental broken symmetry. Most of us have our heart on the left-hand side of our body. Some people actually have the heart on the right-hand side and are perfectly fine. Some people have the heart on the right-hand side and are not perfectly fine. But I want you to notice the journal where these pictures come from, the Annual Review of Fluid Mechanics. That already tells you something. So the fact is that each of us have at least 3 fundamental broken symmetries: top, bottom, front, back, left, right. And the left-right one is the last one to break during embryo development. And it's the understanding of that which is one of the great stories in biology in the last 15 or 20 years or so. And it works something like this. By the way, I should say my computer has chosen this morning to misbehave. So hopefully it won't get stuck. But if it does, please bear with me. In the developing vertebrate embryo, such as this mouse embryo, there is a small chamber called Hansen's node, or more generally, when talking about all sorts of organisms, it's called the LRO, the left-right organizer. It's a small chamber a few hundred microns across with a kind of membrane on top, a sort of pancake-shaped region filled with fluid. And at the base of that chamber is a whole collection of microscopic hair-like appendages called cilia, which are like the flagella that power motility in microorganisms, but these are rather short. And as you see in this high-speed movie that's been slowed down, they whirl around in a very characteristic way. And in so doing, they set up a circulating fluid flow inside this chamber, with a definite handedness in such a way that, and this is still a bit mysterious, the fluid flow interacts with chemical messengers to break the left-right symmetry of the developing embryo. And we know this is the case because the experimentalists who discovered this took the lid off the container, the chamber, and mechanically stirred the fluid the other way, and the mouse ended up with its heart on the other side.
Quick question, what makes this cilia different from conventional ones and deep?
They're rather short, and so they're much more rigid, and so they actually whirl around in a characteristic way. They also have some, the microstructure is a little different, although I forget the precise details. It's slightly different than the usual one, but not fundamentally. Well, if that doesn't answer the astrophysicist as to whether fluid mechanics is important in biology, then I give up. I mean, this is really central to all of us. Now, it's also the case recently that it's become clear that many of these issues about fluid mechanics, about cilia, and about transport have implications beyond just physiology, but also in terms of evolution. And one of the big questions in biology, I would say the number 3 question in biology, number 1 being the origin of life, number 2 being the nature of consciousness, number 3 is the origins of multicellularity. That is, why did the first microorganisms that first appeared on Earth out of the primordial soup eventually transition not from single-cell organisms to multicellular ones that eventually specialized and grew larger and divided up life's functions and became as complicated as we are. And a good way to see that issue of biological complexity is to look at this beautiful graph that was first presented by Bell and Moores almost 20 years ago, and then represented by John Bonner, the great evolutionary biologist at Princeton, who spent much of his 94 years or 92 years worrying about the evolution of biological complexity. So it's a very simple graph. You could argue a bit about the details, but basically for many different types of organisms in the animal and vegetable world, it's a log-log plot of the number of distinct types of cells found in that organism, and you can quibble about how you count, but it's some kind of consistent counting, as a function of the total cell number in the organism, right? So a bacterium, which is obviously one cell, has one cell type that does all the functions of life. It sits down here. Green algae start having 2 cell types or 3 cell types, and by the time you get to a highly developed organism, like a human, we have about 10 to the 14th cells, because we're about 1/10 of a cubic meter, and a cell is about 10 microns on a side. And we have 210 distinct cell types. Now, if you're a statistical physicist, you look at this and try to find a straight line. I wouldn't suggest doing that. It might be interesting to say there's some kind of very low power law here, but frankly, who cares? The main point is there's a general trend that as things, sorry, did I offend anyone? Right. as things get larger, they tend to get more complex, although rather slowly. So if we're going to try to make some progress in understanding this trend, one possibility is to be a bit reductionist and to look in the lower left-hand corner to try to understand, for instance, as we increase the number of cells as an organism and we find the first transition from 1 cell type to 2, maybe we can make some progress in understanding that kind of question. Now, as you probably know, since I know there's a mix of experimentalists and theorists and physicists and sort of biotypes here, anytime you ask these kinds of epic questions in biology, there's a model organism of some sort that is used to answer it, right? So if you have questions about neuroscience, maybe you use C. elegans, and if you have questions about development, perhaps you use Drosophila, or perhaps you use something else. But there's a well-defined small set of organisms for this kind of question. And over the last 10 or 15 years or so, it's become clear, actually, that for questions of this type, green algae turn out to be the model organisms of interest. And this is the title of a review article that I recently wrote in that very same annual Reviews of Fluid Mechanics. It kind of summarizes all the things that you can do with green algae when it comes to fluid mechanics. So I'm going to tell you a little bit about the issue of studying the evolution of multicellularity, different species that build up to become larger and larger structures. There are interesting fluid mechanical problems involving hydrodynamic bound states of swimming microorganisms. There are whole interesting questions about what are the flow fields around these organisms driven by thousands of flagella or cilia around them. The synchronization of those flagella, a very interesting stochastic nonlinear problem of coupled oscillators, basically. Issues about how microorganisms that don't have a central nervous system but nevertheless have a very large number of independent components manage to do things like steer toward the light, as in phototaxis. And there's some physicsy issues about tracer particle dynamics in suspensions of active particles. So there are many things that you can look at using a class of green algae that I'll tell you about. Now, the sort of original hero of today's talk is one Antony van Leeuwenhoek, who was the perfecter of the microscope, and whose original microscopes look something like this. They're sort of large postage stamp size contraptions with a little screw-like mechanical stage and a beautiful, nearly perfect single lens, only a millimeter or two across. And at the time that he was doing this, most microscopes were complicated compound microscopes with many lenses, and yet with this single lens, which he managed to make in a miraculous way, which he didn't reveal at first, he was able to see things nobody else could see. He discovered so many things, it's kind of miraculous. He discovered biofilms on his teeth. He discovered all sorts of algae, many things. It's not clear if he saw cilia, but he got close. And he wrote many letters to the Royal Society about what he found. And my favorite one is from 1700, and you don't actually have to go to the paper edition, of course, it's all online now. And this was an article about what he found in sheep's livers, gnats, and in the excrements of frogs, which is what you studied at that time. And here he reported a beautiful organism, which in his figure 5 shows up as a spherical body with many dots on its surface and smaller spheres on the inside. And he described how this organism would tend to spin around in a beautiful way. But it remained unnamed until about 60 years later, when Linnaeus, in his great book on taxonomy, in the very last entry in the book, gave it the name Volvox for the characteristic spinning motion that it has, from the Latin root volvere, if I'm saying it right. And if you read Latin, it basically describes this organism moving without pause, not P-A-U-S-E, but P-A-W-S, without pause. But actually what we now know is that it moves through the concerted action of thousands of flagella on its surface. And just to give you an idea of what this organism looks like, here is a real-time movie taken in a tracking microscope that we built in my lab, in which we are watching, in the frame of reference of this organism swimming up through a laser light sheet, at its characteristic spinning motion. The little jitter is the motors of the microscope stage recentering it. And you see tracer particles that move around here. The organism has daughter colonies that grow up on the inside, about 1,000 or more surface somatic cells, each of which have 2 flagella and give it motility. Those flagella beat from the North Pole to the South Pole in a very organized way, but with a slight east-west twist, so the whole thing spins as it swims around a body-fixed axis. And because of the steady motion here, you can average over 50,000 frames of your video and do particle image velocimetry and get extremely accurate flow fields from this, and then try to build theories that connect the action of 1,000 flagella on the surface to the large-scale flow field. Yes?
So it actually has a fixed top and bottom?
Yes, just like you. And me.
What's the size of it?
Yeah, I'm sorry. This is typically about 500 microns across, half a millimeter.
It's going half a body length per second?
Yeah, they can swim several hundred microns per second. Yeah, they're very efficient. Now, they're beautiful to watch. You can spend all day watching them, as van Leeuwenhoek did. But the other thing to say about them is that since about 1892, it's been recognized that they are really interesting model organisms for the evolution of multicellularity. And it was the great biologist August Weismann, who is famous for his work on so-called germ plasm theory, who wrote some very beautiful essays about evolution and all of that, and pointed out at the time why he thought the organism was so interesting and its cousins were so interesting for studying evolutionary origins of multicellularity. So the first thing to say is, well, they exist now. We don't have to look in the fossil record. You can go to the River Cam, scoop up a bunch of water, and you will find Volvox there. And in fact, in the collections of algae that you find around the world, like in Göttingen and Scotland, if you look at the notes for one of the species, it'll say, found in the ditch behind Queen's College, Cambridge. Right, so they're really local. So easily obtained in nature, and now they're being studied from many different perspectives. We know something about their ecology, we know something about their biochemistry, about their genetics, and all of that. And importantly, about their ecology, the niches that they occupy in space and time. Also, because the sequencing is going along, and this organism is not so ancient, a few 100 million years, it's possible to make some comments about what evolutionary transitions gave rise to what features. And there is, from that, evidence of interesting repeated genetic changes, that evolution of multicellularity occurred multiple times, even within the lineage I'm talking about. And it's actually amenable to DNA transformation, although not so easily. Okay, so, well, here's where my computer is pausing. Please? Oh, sorry. Yes, no, it's not pausing. Right, it's spherical. Right, so for those of you whose minds think like a theorist, you know, the fact that they're spherical makes life tremendously easier. Okay, so let me tell you about, first, just to put in perspective, two ends of this lineage of green algae. The first is Chlamydomonas, which is a 10-micron size unicellular photosynthetic flagellated eukaryote. Okay, so it has a little eye spot that allows it to sense light. It's green, it's photosynthetic, it needs to go to the light. It has two flagella, each about 10 or 12 microns long, and it swims with the breaststroke through its fluid environment. It's a workhorse organism for understanding flagella, photosynthesis, all sorts of things. Here's Volvox on a scale where you see its individual surface somatic cells, which are mounted in a transparent extracellular matrix, are basically like Chlamydomonas, although instead of the two flagella going like this, they go like this. And that gives rise to the coherent motion across the entire colony. So several hundred microns across, surface cells at the sort of thick crust of this extracellular matrix, and then inside are the daughter colonies, as you see here, and they grow up to become the next generation. Now, nature's been very kind to us in the sense that we have an entire spectrum of Volvocene green algae, which consists of 2 to the n Chlamydomonas-like cells arranged in some kind of geometric structure. So there's something called gonium, which can have 8 or 16 cells, which is kind of like a flat plate of sorts, slightly bent. And then there are ever larger quasi-spherical structures. You can see the extracellular matrix there. And this goes all the way on to different species of Volvox, some of which have 50,000 cells and are several millimeters across. So we go from 10 microns to several millimeters. That's a big spectrum. And therefore, it's possible to use these organisms to look at the kind of scaling laws for anything you wish, swimming speed, nutrient uptake, etc., to try to understand how nature has chosen to move toward multicellularity. And the important thing is this dividing line. This is an organism like a bacterium that does all the functions of life. It does photosynthesis and it reproduces, and it has 2 different parts of its life cycle, reproduction and everything else. And that's true of all of, excuse me, all of the cells inside each of these species. These are not transient accumulations. These are distinct species. But by the time you get to this dividing line, all the proper Volvox species have 2 cell types, specialized germ cells on the inside and what we'll call galley slaves on the outside, which only do swimming. And the galley slave comment comes from my long-term collaborator, John Kessler, who I think was very clever in coming up with that description. So this is the most elementary distinction in biology that there is. Germ-soma differentiation, somatic cells versus reproductive cells. And so a natural question is, why did nature put this dividing line here? Is there something special about this size or number of cells that suddenly makes it evolutionarily advantageous to break up into 2 types? And you know from any human experience in a group, whether it's a company or a department or a university, that at some point, as things get larger, it's better to become specialized. And here's a perfect test case for this. Okay, so in order to study this in the lab, we use a variety of techniques, most of them borrowed from the world of in vitro fertilization or the macroscopic world of fluid mechanics experiments. Here, for instance, is Volvox on a beautiful micropipette. This is a glass capillary that's been heated in the center, pulled really fast so it necks down to a tiny tip, and then heated again to round up. So you've seen pictures probably of in vitro fertilization where an egg is being held and a sperm is being injected or something like that. It's the same technology. And if you suck on this a bit, you can even deform the extracellular matrix. And so with this, you can study many aspects of these things, the fluid flows they create, the synchronization of the flagella. Now, for the purpose of today's talk, I want to show you the life cycle of this organism because it's very important for what I want to say about cell sheet dynamics. So in the lab, we typically grow this on a 48-hour life cycle in a diurnal growth chamber. It's a big box like a refrigerator with a temperature and light control for day and night. So here they are growing. And if we pick up the life cycle at the beginning of the first morning, These daughter colonies, shown as small spheres, hatch out of the mother, and they have inside of them small, undivided germ cells. And then during the first day, those germ cells grow very large before dividing at all, and then rapidly go through 9 or 10 divisions to produce something like 2 to the 10 cells of the daughter colonies. Now remember, these organisms swim with flagella that point out, and they have inside of them, as an adult, the germ cells. But when these daughter colonies are first formed, they are inside out. They have their germ cells on the outside, and their flagella, or the places where the flagella will emanate, pointing in. So it's just wrong. And so during that night, they go through a process called embryonic inversion, where they turn themselves inside out. And here are some electron micrographs showing you this process. I'll say much more about it shortly. But basically, with no assistance, they manage to turn themselves inside out, invert the sphere. Now, they cheat a little because there's a little hole at the top to begin with. You may know the famous math problem. Oops, why did it do that? Where you have to invert a sphere without singularities. Okay, and then during the rest of their life cycle, all that happens is that the extracellular matrix gets expanded and the cells, the somatic cells, move further apart, but there's no more cell division. So during this later stage of development, the only thing that's happening is a slow march through parameter space. The size changes, the spacing between the flagella changes, the density offset changes a bit. And so you can use that to study many physical properties as a function of size. Yes?
When this cycle are the new germ cells generated?
So in that, when I said there were 10 stages, 10 cycles of division from the initial germ cells, at that moment, at the end of those 10 cycles, one has the next germ cells and all the rest of the somatic cells.
This is before inversion?
Before inversion. So they're on the outside. They're produced and arranged on the outside, and then they have to flip to be on the inside.
Question, apparently using the term extracellular matrix somewhat different from what I use.
Okay.
So what is extracellular matrix in your case?
It's a complicated polymeric cross-link network. It's transparent in the visible.
On the surface of the...
Yeah, well, it's actually got some thickness to it. So it's kind of very dense at the surface and more tenuous on the inside. I think a good analogy would be the inside of the whole organism is a bit like the inside of your eye. So it's more viscous. There's stuff around, but it's very thick right near the edge. So, for instance, if you want, you can measure various things like the speed with which the organisms swim up against gravity as a function of their size, where size you could read for time through that second day. And so when they're young, they swim up very fast, and as they get old and heavy, they have some trouble. If you deflagellate them, you can look at the speed with which they settle under gravity, and from Stokes' drag, you can figure out the density offset. It's basically a linear function of radius. And remember, they got their name because they spin, and that spinning frequency is a very strong function of radius, so it's very high when they're young, and it goes to probably an order of magnitude lower by the time they're quite old. And then because these organisms have their daughter colonies in their posterior, they are bottom heavy, like most of us. And so if you tilt that axis away from vertical, there's a gravitational torque pulling them back, and it takes about 10 seconds for them to reorient. And you can measure that as a function of time. And so you can pick and choose what you'd like to study. So for instance, if you want to study some of the guys who are very fast swimmers and don't really care much about gravity, you could put them in a box with a glass top on it, and you can watch that they swim up to this glass ceiling, and they do this. They spin around as they naturally do, but when there's a perturbation, as there will be, this guy swims past, this one is pushed away, and then it comes back. So there's actually an attractive interaction between these two rotating organisms near an upper no-slip surface. And you might think about chemotaxis or all sorts of things, but no, it's actually purely fluid mechanical. And it can be described parameter-free in terms of two interacting Stokeslets near an upper wall through that no-slip boundary condition using the famous work of John Blake on Stokeslets near a wall. And so it's a beautiful testing ground for all sorts of interesting issues in fluid mechanics. Okay, so again, the technology we're going to use for that kind of investigation involves high-speed cameras, inverted microscopes, and micropipettes held on micronally accurate micromanipulators. And when you use that kind of technology, you can see, for instance, the beautiful beating of the flagella on one of these surface somatic cells. So here's Volvox on a micropipette. Here's a daughter colony. It's been image processed, obviously. So this whole thing is 0.5 millimeter across. This is about 12 microns. And you can see the beautiful beating of the two flagella of the somatic cell and some sense of coordination with the others nearby. So that's the way they ought to be. And I just want to read that into the record that we're talking about motile organisms that have to have their flagella on the outside. Yes?
At this stage, are the germ cells inside?
Yeah, this is an adult, so the germ cells are inside. Okay, so now I'm going to segue, just 1 second. I'm going to segue into the real purpose of today's talk. Yes?
Are these two flagella beating independent of each other?
That is a great question. Yes, I put this up precisely because it's confusing. Sometimes it appears that they are synchronized, sometimes not. They could be two oscillators with slightly different frequencies that don't talk to each other, and like any nearby oscillators in frequency space, they'll occasionally line up, or they could have synchronization. So actually, that's a whole other lecture about how they synchronize. And this is actually the unusual case. Most of the time, the two on a given somatic cell are pretty highly synchronized. Okay, so there's a famous quotation that I want to tell you about. I don't know if any of you have heard of this before. It is not birth, marriage, or death, but gastrulation, which is truly the most important time in your life. How many of you have heard this? A few, okay. So I knew about this somewhere in the distant past, and it's due to Lewis Wolpert, the famous biologist, and his son is a professor here. So I emailed Daniel Wolpert. I said, Daniel, what's the reference? Or can I get in touch with your father? I didn't have his email address. I'm trying to find the reference for this. So he connected me with his father, and I said, so what's the reference on this? And he said, actually, there's no reference. It's all apocryphal. He said, I was talking to a doctor once, and I was shocked to discover he didn't know what gastrulation was. And there was a friend of mine there listening to this conversation, and then I said this, and the friend thought it was so clever, he wrote it down. And so it doesn't appear in any literature, primary literature, but it's now been in the literature enough because people repeat it that we all know it. So it's a beautiful statement about a very important developmental stage in biology. So just to be clear, gastrulation is that process in early embryonic development in which an initially spherical mass of cells, such as you see here, develops an invagination that eventually leads to the formation of the gastric system and eventually to a change in topology from that of a sphere to that of a torus with a hole all the way through, like we all have. Now, gastrulation is a beautiful example of a complex process in biology that changes topology. And we now know that there are a whole bunch of different physical phenomena or biological phenomena going on as this occurs. There are cell shape changes. There is a certain amount of cellular differentiation, cellular migration, and cellular intercalation, so that you have multiple differentiated layers of tissue that find their way in here. But nevertheless, if this doesn't happen, you're not here. And that's why Lewis Wolpert said this is really the most important time in your life. But these kinds of cell sheet folding events are all over biology. Another example is in neurulation, where the spinal cord region is formed by the pinch-off of some tissue, as you see here, that leads to a kind of topological change, leaving the spinal cord region separated on the inside of this organism. So there are many, many examples you can find where these kinds of closure or topology changes occur, and I'm now going to tell you about how a particular process, that process of embryonic inversion in Volvox, is a very simple model system for understanding this because it only involves one of the many processes I just told you about. Now, let's see if my computer complains. No, good, okay. So in Volvox, there are two types of embryonic inversion, and they are known as type A and type B. In type A, a cross-shaped opening, which is called the phyallopore, Greek for bowl and pore, opens at the top of the embryo, and 4 lips peel back, initiating a process that is schematically shown here that results in inversion. In type B, there is a circular phyallopore at the top from the beginning, and an equatorial invagination that moves the posterior up into the anterior. The phyallopore stretches, and then eventually the system inverts. And you see videos of this happening here. These are ordinary light microscopy videos. Now, this is a kind of miraculous process, and a lot is known from the biology community about what's going on. There is one particular species that shows something that's sort of intermediate, where you get 4 lips and an equatorial invagination. But anyway, let's not worry about that detail too much. What we do know is that if you take, for instance, Volvox globator, which is shown here, and you look at this type B inversion, that if you stop the process somewhere in the middle, fix the organism inside a resin, slice it with a microtome, look at it under the microscope, you'll see that in this bend region, the cells have actually changed their shape. So what were sort of squat ellipsoids become elongated, sort of paddle-shaped. But secondly, that there are cytoplasmic bridges that connect one cell to the next. These are left over from cell division, the kind of incomplete cytokinesis. And that's what's holding the whole thing together, because at this point there's no extracellular matrix. And whereas elsewhere in the organism at this point, the cytoplasmic bridges are sort of at the equator of this line of cells, here they migrate to the tips. And so imagine taking bowling pin-shaped objects and cinching them together at the tip. They will splay. And so that confers a preferred curvature to the cell sheet and starts this process. And we know this from these kinds of studies. We also know it from various kinds of electron microscopy on fixed embryos. These are gorgeous, gorgeous pictures, obviously frozen in the middle of the process, different embryos. You can see this mushroom-shaped structure where the posterior is a bit contracted. And then over time, these cell shape changes spread over the entire body of this organism and make inversion complete. So the picture really is that this is a series of, let's say in some sense, equilibrium structures where time is just the progression of this wave of cell shape changes, and that accomplishes inversion. That is the cartoon that has come about from these kinds of biological studies. And we know, for instance, through the study of mutants, which are deficient in certain motor proteins that I'll mention in a moment, that the two processes of cell shape changes and migration of the cytoplasmic bridges are both needed in order for this to occur. So, for instance, the migration of these cytoplasmic bridges is accomplished by a motor protein that is walking along microtubules inside the cell and actually pushing the cell relative to the bridges, and that's what's giving rise to this change of connectivity. And so if you have a mutant that's deficient in that particular kinesin, then inversion doesn't complete. But actually, perhaps the most interesting thing, I think, from a physics, mathematics type audience is that there's a whole spectrum of ever more complex cell sheet folding, it's really hard to say, cell sheet folding events as a function of size. So if you go to gonium, this plate-like structure organism that I told you has 8 or 16 cells, it just goes through a kind of wimpy flip. So the curvature just flips from this to this, like an ammonia molecule during the course of inversion. But as you get to larger and larger organisms, you find ever more elaborate cell sheet folding. And by the time you get all the way to the ones I've been talking about, like globator, then you get this beautiful type B inversion. So there's a hope here that out of all of this, there would be some systematics from elasticity theory and all of that about the size dependence of these structures. I think that the open questions, which are basically all the questions you could possibly ask about this system, are first of all, let's focus on this type B inversion. What triggers the beginning of this? How do the cells know in the equatorial region that they should start doing this process? Is there a morphogen? Is it some kind of elastic stress? Is it just hardwired from cell division? We don't know. There's been a lot of discussion in the literature about a role for active contraction or expansion of these cell sheets in addition to cell shape changes. We're looking to understand eventually systematics with colony size, and then eventually issues about how this extracellular matrix is added. Question, yes?
Maybe this is naive, but is there any way to measure energy that is being consumed in this process?
I don't know off the top of my head, but there might be some measuring would be perhaps different than controlling in the sense that maybe one can change the amount of ATP that's around in the cells or something like that. Of course, you can also imagine going in and cutting these things, as some people have done, in the middle of the process to see if there is residual stress that's built up and then see what happens. So that gets a little bit to what you're asking. But I don't know of a direct way to do it yet. But that's a great question. Okay, so our starting point here was to say, look, the experiments have to lead here. What we really are lacking in this system is a quantitative three-dimensional view of what's happening. And hopefully we'd eventually be able to track every single cell and understand the shape in 3 dimensions, and from that be able to use what's called the scientific method, right, to compare theory and experiment. But from light microscopy, it's not so easy, unless you're really lucky, to get a good image of what's going on.
I have a biological question.
Sure.
What gives the cell the shape? On the cartoon, it was a microtubule. So is there a microtubule cortex, or is there?
There are microtubules that are running along the length. Yeah.
They give the cells their shape.
I'm not sure if I would say they totally give it their shape, but certainly in these elongated structures, there's a parallel bundle that's playing a big role. Yeah.
Do you know something about actin? How it is distributed in these?
Actin plays a role. I don't know off the top of my head how much of the shape is precisely driven by actin, but actin plays a very important role. Yeah. Okay, so I want to tell you about an emerging technology. We are not the inventors of this. We are just users of this technology. It's called selective plane illumination microscopy, SPIM, or light sheet microscopy. It's becoming widely used now, and it's a technology that is perfectly suited to this. So you're all probably familiar in one way or another with confocal imaging, which has great meaning very small, depth of field, and from which you can take multiple slices of a biological sample and build up a 3-dimensional stack of images. That idea of a stack is great. Confocal imaging is not capable of dealing with organisms of this size. There are fundamental limits to the confocality in most confocal microscopes, and you can't go beyond 100 or 200 microns. But light sheet microscopy is basically a technology in which you have a laser somewhere, you make a light sheet out of it, you send it backwards through a microscope objective to make a very small laser sheet, and you send it through your sample, and you can translate the sample very precisely through the laser light sheet and collect orthogonally to that fluorescence of one sort or another coming from the sample and build up a 3-dimensional image that way. Now, this process of inversion takes about 45 minutes to an hour, and you can take one of these stacks in a minute or two, so you can really get a nice snapshot of what's going on. The nice thing is that there's this wonderful place, openspim.org, where you simply get a parts list to build one of these microscopes. So it costs 15,000 pounds or something like that. Well, a lot of that is the objectives and the camera and things like that, but it's not so bad. And it's not only a parts list, it gives you everything. You know, for those things you can't buy, here are the drawings, and here's a little movie to how to put it together. It's great. So I highly recommend it. And you can find some of our contributions there, but as I said, we're just users.
Question to this.
Yes.
How do you fix your Volvox in the focus?
Yeah, so one way we've done it is actually to embed it in a very tenuous agarose medium, so kind of like a gel, and then just extrude it from a capillary, so it's hanging there kind of gently held, yeah. Okay. Or it can be on a micropipette. So this is what the thing looks like. It's about this big, and so we pick off a laser from down below. It comes through optics to produce that laser light sheet. It comes into a little chamber which has these 2 objectives. Now we actually have 3. We have light coming in 2 directions, and we look orthogonally, so there's no shadowing. Here's the sample chamber. The objectives are water immersion, so they're just in there with the Volvox. And then it goes out to a camera. And if you look closely at the little chamber, it's just made of plastic and watertight, and everybody meets right in the center of the 2 objectives. So it's a wonderful little system. And here, I have to make a pause. I brought 3D glasses.
Oh, good.
Yeah, so please take a few and pass it around. I'm going to show you, yeah, I should have done that earlier. I'm going to show you some movies of this process, both looked at from afar, where we see a single mother with multiple events happening, and then we'll zoom in on one particular inversion. Maybe if we take a bunch and distribute. Can someone do that? Yes. Too much multitasking. Just take 10 and pass it to the next row. Yeah. It's one of those linear programming problems. How many physicists does it take to distribute?
Usually quite a lot.
Okay, so while that's happening, let me first show you something that doesn't involve a 3D movie. Okay, so here's a stack of images through a Volvox colony. This is not 3D. So you see we're just slicing through, and every once in a while one of the daughter colonies shows up like a firework. And you see it's mostly empty space, right? But as you get toward the other side, you see just at the last bit the kind of hexagonal network there. So,
What's being illuminated?
okay, so what we're using here is autofluorescence of chlorophyll. So these are all photosynthetic organisms. You shine a blue laser on them, and chlorophyll fluoresces very nicely, and that's what you see. So you don't have to add any sort of dye, although you can if you want to see other things. So the next movie is not 3D either, so you still have time. This is a slow movie. Slow, well, this is a time-lapse movie in which you'll be able to see the subsequent successive inversion events of each colony. Now remember, these things have flagella, and when the flagella point in, it's not going to swim. But as soon as they turn around, the flagella are on the outside, and it starts spinning. So, boom. This one's, well, you know, we're taking movies every few minutes here, so there's a bit of strobing effect. But what you see is, inside the mother, there are multiple daughter colonies. They're rather large in globator. And there's this inversion event. There's some interesting fluctuations. Then they turn around. So it's not perfectly synchronized, but there it is. Now, you all have glasses? Okay, good. Here's let's hope this works. Here's a time-lapse movie. You can see the mother growing. Can you all see that? Yep. And the individual events. And so actually there are many, many interesting things you can do with this. And at the end I'll mention this business about actually tracking the individual cells on the surface and looking at how they're organized. But this is the raw data in a sense. Now we're going to zoom in on one particular embryo. And this is going to go by too fast to see, but I mean, you'll see it, but you won't really understand it. But here are the germ cells of the daughter, and the whole thing just went like that, right? That took 45 minutes. So you see there's a hole at the top, and then whoosh. Okay. So now let's look at that more slowly. So you'll see there's a bit of denting, as we call it. There's certain weird fluctuations that occur. And then you see this beautiful mushroom shape come in, and the whole thing flips around. Is that clear? Yeah? Okay. So, yeah. So I submit to you that this is the world's simplest example of cell sheet folding. And if we can't understand this, we're not going to understand gastrulation, I think. Okay. And then I think that's the last of the 3D movies. Here are some other examples of this process. It's very repeatable. You have this beautiful posterior moving up into the anterior, and then everything slides around.
Does the opening happen when the bottom part?
It's always there in a small way, but it really starts expanding when the posterior starts moving up.
Okay, so it's separate events.
There, it starts expanding.
Yeah.
Okay, so the point here is that any one event is fascinating, but if we're going to really be able to understand what's going on, we need to be able to study tens, scores of events, do some statistics, understand what the typical behavior is. And because we have this stack of images and we have access to modern image processing software of the kind that's used in medical studies, for instance, we can take this stack of images and slice it and dice it any way we wish, so that we can get an accurate measurement of the overall symmetry. We can measure the surface area. We can measure all sorts of things. So for instance, here are some stages where we take cross sections. We can then image process a kind of average contour. We can measure the curvature along the surface, and we can measure the surface area, assuming it's a surface of revolution. And we can measure something. And by the way, there's no guidance in the literature about what to measure, because nobody's ever thought of this before, really. But we've been looking at the distance between the posterior pole and this tight bend region as a kind of reaction coordinate, a measure of how far along we are in the process. We'll call that little e. So here's what you can do. This is that quantity e, non-dimensionalized by its initial value, as a function of time for a bunch of embryos, where we observe that there is a noisy evidence for a kind of speed up of the rate of motion of this posterior position, and we've redefined the zero of time to be there. So we're talking about something that takes about 45 minutes or an hour, and we're just going to call that speed up time 0. And this characteristic speed up has been seen once before in the literature in a kind of anecdotal study of type A inversion, but it seems to be robust.
That's also you define E naught?
E naught is just what we measured initially at some particular time.
Is it relevant that it's 1?
That's by definition.
Okay, yeah.
Yeah. Well, actually, it's by definition at something like this point in time, and then it goes a little back. So there's a bit of uncertainty about how to do this. But you know, we pick a time and say, when we can first measure things, let's call that E naught. Now you can actually measure the surface area of the entire entire organism as a function of its initial value. Again, as a function of time, there's some indication for a slight decrease, then a very large increase, and then a very large decrease in the inverted state. And then you can measure things like, say, the maximum curvature in this bend region, which is negative by definition, as a function of time, and it peaks negatively right about time t equals 0. But the thing I want to point out here, which is very important from a theoretical point of view, is that I've told you that there's strong evidence from the biology that there are cells that have changed their shape in this bend region, imparting a preferred curvature, and that's what's starting to drive it. But there's a second bend region right here. If I didn't tell you that first part, and I said, well, somewhere there's an intrinsic curvature to this cell sheet, you wouldn't know necessarily that it should be there or there. When I have a three-dimensional closed object with the topology of a sphere, I can't just trivially look at the shape and say, I know where the preferred curvatures are, because there are global constraints on being closed. It's a lot different than if I had a filament, like a hair, a long hair, that might have built-in curvature. With the exception of the occasional self-touching, I could look at it and say, oh, I see the preferred curvature, and that's the actual curvature. But you can't do that for these kinds of objects. So it's important that we have the microscopic information as well. Okay, so now I'm going to talk a little bit about how you might model this. And our starting point for this was actually the theory of fluid membranes, which probably many of you know about. So if you go back to the original work of Helfrich and Conham, who were worrying about the shapes of red blood cells, erythrocytes, the so-called biconcave discoid shape, there was proposed a model which really derives from elasticity theory of thin shells and plates, but for fluid membranes it's got a slightly different structure, in which there's an energy functional that's a surface integral of a quadratic form in the curvatures 1 over r1 and 1 over r2 that you could ascribe to every point on the surface. So from this you make the mean curvature, the sum of the two, and the Gaussian curvature, the product. So up to quadratic order, you have a term in the Gaussian curvature with its own modulus, and then you have in principle not only the preferred curvature, but possibly some constant, or even could vary in space, called the spontaneous curvature, and that quantity is squared. Now, the surface integral of the Gaussian curvature over a closed object is a topological invariant, and so if you're just talking about changing shapes, that's a constant and not interesting. But it's this idea of a spontaneous curvature which has played a very important role in the theory of membranes and related systems. So for instance, a kind of crude way of understanding the biconcave discoid shape of the red blood cell is to say it's trying to minimize the integral of the square of the mean curvature subject to the constraint that the surface area is given and the enclosed volume is given and is less than the equivalent sphere. So as you know, with a football or any inflatable ball, if you have some missing air in there, there's going to be a dimple. It has to be somewhere, and minimizing the energy will put the dimples symmetrically on either side, and you're done, except that it's wrong, because if you wash away the membrane with a detergent, you still have the same shape, because it's imprinted in the spectrin network, which lies underneath. But anyway, you could still believe that that was dictated by elasticity.
You need your surface to be well connected?
Well connected?
Yes.
Meaning what?
For your biological problem, the two ends, some ends are not, the surface not completely well connected.
This is true. So there's a tension theoretically here about when can I get away with using a continuum theory for something which is really a bunch of cells held together with little thin membrane tubes. And we're going to explore the possibility of doing that, but we're mindful of the fact that it's not so necessarily well connected everywhere, and it could be a problem. So yes, that's one of the things I was hoping to sweep under the rug, but now I can't.
Can I also ask something? So given the volume and surface area constraints, do you really need spontaneous curvature?
Well, wait. All I've said is this is the starting point to talk about it. I'm not going to invoke any volume constraints in this model. I'm just saying that if you kind of read the textbooks about how this might relate to this, that's what's said. But you can poke a hole in this, and it still has the same shape. So actually, it's much more complicated than just saying it's an energy minimizer of this by
Well, I
itself.
was more saying that if you minimize this term, you might not need a spontaneous.
No,
curvature
absolutely.
to
You
get
might not need it at all, right? But it can produce interesting things. But it was introduced as a concept there. Now, the origins of spontaneous curvature from the point of view of membranes are many. You can have different lipid compositions in the two halves of the bilayer. So you can have bigger head groups on one side than the other. You could have proteins that like to gather around on one side of the membrane and not the other. You could have different charges on the two sides of the membrane. You could have protein scaffolding that comes along, proteins that insert preferentially, all sorts of things that could break the symmetry. So let's see how we would apply that to our problem. Now, I'm not going to show too much mathematics here because I just want to give you an idea of where we go, but I will show one slide of mathematics. And as someone I know once said at Bell Labs, keep the mathematics simple for the benefit of the theorists. I include myself in that. Okay, so the essence of this model is to just use the theory of thin shell or plate elasticity, kind of like the Helfrich model, but done for something that's not a fluid. It's an elastic solid and has to be treated as such. And the basic idea, which has been discussed a lot recently in beautiful work, for instance, by Alain's colleague Dominique Vella at Oxford, work on indenting pressurized objects like beach balls. You know, you push and you get a beautiful deformation and you try to understand the mathematics and physics of that. There are lots of interesting problems in physics and biology where this kind of description matters. And the basic idea is to say, I will describe the energetics of my system in terms of some reference system like a sphere where the geometry is simple, and I'll talk about the deformations that take one to the other. So again, the hypothesis, which we are really just going to test here, is that a quasi-static sequence of shape transformations of cells translates into intrinsic curvature, stretching and contraction of an elastic sheet, and that might explain what's going on. So the basic idea is, as you see, you have a reference system which has its own internal arc length variable. Let's assume rotational invariance. It has its own tangent angle. It has a radius as a function of position. And then my new shape has its own arc length variable, S, and its own tangent angle. And I can do two things. One is elementary geometry that relates the radius, the arc length, and the tangent angles. But the other thing is to say that the rate of change of one arc length relative to the other gives me a stretch in one direction, and the ratio of the radii of the initial and the final state give me an idea of a stretch in another direction, and those stretches lambda, as they deviate from one, give me strains, and I'll form a quadratic energy functional out of those and out of what are called curvature strains, which are measures of the local curvature, kappa, in the two directions, azimuthal and meridional, relative to some preferred curvature that might exist. So it's a quadratic energy functional, just like Hooke's law. There is an assumption that there's just a uniform elasticity in the system governed by Young's modulus. H is the alleged thickness of this cell sheet. Nu is the Poisson ratio. The bending terms come in higher order, H cubed, and have a definite structural form. And the question then is, what sequence of preferred curvatures and stretches or contractions might give rise to what we see? So we're going to try to find energy minimizers of this under that statement. Yeah?
The system is anisotropic in your model, or isotropic.
Isotropic.
But then the spontaneous curvature should be probably equal in two directions.
They're not.
Kc and K.
No, but sorry, I thought you meant the elasticity. No, the spontaneous curvatures can certainly be different in the two directions.
It's anisotropic.
Yeah, sorry, I misunderstood what you meant. Yes?
What's kappa? What's kappa? The kappa.
The local curvature. So one is the preferred curvature, one is the actual curvature. Okay?
Going back to the two issues. Back to anisotropy. Is there a biological reason to think that the cell layer is anisotropic?
Yes, actually, when the cells become paddle-shaped, they're actually slightly different when looked at in the two directions. So you can imagine anisotropies from that. I mean, these are subtle points, but yes.
But that means that you have some kind of pneumatic order of the cells in the plane, which is, in principle, another degree of freedom that you could add to this. In other words, if you assume they're perfectly, they're elliptical and they're all aligned in a certain way, then you'll get two spontaneous curvatures, as you told Misha. But the fact whether they are aligned or not aligned could also vary with shape.
It could,
That could be another
but
degree.
one thing to remember is that all of them have roughly 6 connections to their neighbors of these cytoplasmic bridges, which are kind of constraining that alignment a lot. I mean, you could agree, I guess you could say, well, if they're not symmetric rotationally, each individual cell, then I could have some internal discussion about how they arrange. But that, I would say at the moment, that's a level of complexity that would be way beyond what we could be sensitive to. But in principle, yes.
The idea is that you prescribe this alignment and out of that you get the curvatures, and that's given for your theory.
Yeah.
So you've done something you solve for, it's not a
Right.
dynamic environment.
Right. So my motivation in coming up with the simplest picture may sound a little bit strange, but let me tell you. I remembered when I learned statistical physics that if you want to try to understand things like virial coefficients or any properties of a non-ideal gas, well, there's a whole Mayer cluster expansion where you put in the intermolecular potential and you turn a crank and out comes the answer. Now you can put in the complicated intermolecular potential between two argon atoms or whatever, or you can just put in a square well potential, infinite here, finite width of a constant energy depth, and then nothing going on, and you get basically the same result. The essential physics of the problem doesn't care if you replace the complicated interaction by something piecewise constant. Now we know from the biology in this problem that there seems to be a narrow region where, initially at least, there's a preferred curvature and not much happening elsewhere. So let's just adopt that point of view, not try to fit some complicated form. We'll just assume that over some small region, lambda, of width lambda, that the preferred curvature just dips to something negative and then comes back to what it was before. And see how far we can go with that. And you'll see we can go pretty far. Now, the other thing is that we have evidence from the imagery, even before our own imagery, that the posterior, for instance, at some point contracts. And this is believed to actually involve an actin-based process in which the cells elongate but keep their connectivity at the equator. So if the cells elongate, the whole sheet contracts. So there's the possibility of imposing a stretch or contraction in the system. Actually, there's some evidence that the anterior may expand as well. So we can put in these things as we wish to try to test what are the essential features to make inversion work. And then the question is, what are the energy minimizers of an elastic shell endowed with these preferred curvatures and stretches? So here's a little illustration as we crank up the magnitude of these curvatures of a couple of essential points. Imagine that you just take this spherical object and produce a preferred curvature that wants to bend inward at roughly the equator, at the waist. What will you get if there's no contraction anywhere? Versus what happens if you contract your posterior, or if you misplace where you put that preferred curvature because you think, because of the bend, that it's actually somewhere else. So the first thing you see as you increase the magnitude of this curvature is a purse string effect. Right? It will cinch it in, that's fine, but it can't go any further. But if you add posterior contraction, then it's possible to actually push the posterior up into the anterior and get much further along in this process. On the other hand, if you put the preferred curvature a bit above the equator and made it bend inward in the way that you see it here, then you get this smiley face, and it's certainly not going to invert. Now, I can't prove by rigorous mathematics the kinds of things I just said, but I think it's intuitively clear that there are certain things that will work and there are certain things that won't work. And in order to get the posterior to fit into the anterior, it's necessary, it seems, to have a combination of this inward preferred curvature and posterior contraction. And that was something that the biologists had suggested was important as well.
Are these simulations that
So this is a time series in which the magnitude of these effects was quasi-statically changed, and we found the energy minimizer at every point. So there's no real time here, it's just a sequence of changing the parameters, the depth of the preferred curvature, and in this later point, at some part after we bent in, we added posterior contraction.
It's a gradient flow.
Yeah, but time is just a parameter for the magnitudes of those effects. So we started thinking, okay, maybe we can actually be a bit quantitative here. We've got all these embryos at the initial stages. Now they're different sizes, so we can trace their contour, we can rescale them to lie them on top of each other, and try to pick a point in their evolution, maybe as measured by this little E parameter that tells us how far the posteriors move, to say, okay, these are all at the same time, or same stage, and then average them. And we get, for instance, this graph with its error bars is the average shape of an embryo at this point in time. And then we can ask, okay, can we fit it? Can we fit it with a region of preferred curvature of a size that's what we know to be the case from the thin sections? And the answer is yes, within the experimental error. Now, as you go to more complicated invaginated shapes, you can see it's much more variable. There's still an average quantity, and you can still, with a bit of more work, you might have to fiddle with expansion and contraction, you can actually get a reasonable fit to that as well. But it requires understanding that there's this preferred curvature localized in this region. Now, if you go further along in the process, this is now the latest results in which we have many, many embryos, and we've averaged over all of them. The black shows the average shape, and as you see, there's a pretty well-defined average process of posterior moving up and the rest folding back. And by fiddling with these parameters, you can actually get a reasonable accommodation of the whole thing. Now, if you have expansion and contraction and this preferred curvature as a wave, so that it's actually enveloping the entire posterior, then you can make a first attempt, which is what I've shown in snapshots there, at the entire inversion process. So the contraction, the expansion of that wave of preferred curvature, and then it fills up the entire shape, and you do invert the sphere. So I'm not trying to say, you should believe every little detail of this, but it's a spatiotemporal scenario of contraction, expansion, and preferred curvature, which accomplishes what we see in biology, in the experiments. And it's at least a candidate idea for how biology does it. Yes?
Just a question. Do you really need contraction, or you can manage with the interplay between two curves?
So it seems that posterior contraction is really important and essentially necessary for this to happen. It's aided by anterior expansion. And we know from electron micrographs and other studies that there appears to be a rearrangement of the cells in the anterior that essentially corresponds to expanding the cell sheet. They change their orientation and their connectivity a bit. So there appears to be both of these, and if you incorporate that, it becomes that much easier for it to work.
So at least looking at the images I show, it seems apparent that the main difference between the experiment and your simulation is that this posterior area is not as circular as an experiment. So what makes this deformation like what's...
Okay, so this gets to a very interesting question. One of the thoughts we had initially was, how can one distinguish between what you might call passive contraction or expansion versus active contraction and expansion? If you put, the nature of the elastic problem is that if you put this preferred curvature somewhere, it's going to spill over to nearby regions in terms of what the cell sheet does. And there's some kind of decaying, oscillating, sort of exponential length scale on which that disappears. But the region of contraction that we see is much larger than that, suggesting that it's really active contraction. Now, whether that active contraction is perfectly homogeneous over the entire posterior or not is anybody's guess. And so if you want to get a better fit, you have to do things like make a graded contraction or graded expansion. So we can do that, but it's not particularly exciting because you're just sort of playing with parameters.
Is the value of the spontaneous curvature that you need to sort of understand this of the order of the inverse of the cell thickness? Somehow I'd imagine that the spontaneous curvature should have to do with the difference between the size of the top and the bottom of the cell. And somehow that should be the cell.
It's actually consistent with that. Yeah,
Okay.
it's consistent with that. And of course, it's also, unfortunately, as Alain would remind me, I think, it's also in a regime where the assumptions of thin shell elasticity are probably no longer valid, right? But we're trying to just push this to see where we can go.
What is the meaning of the red region?
So the red region is the region in which there is preferred curvature.
But so that's a constraint. I mean, that's saying the spontaneous. If we were, say, talking about the Helfrich model, it would mean that you would the region which H0 is prescribed is changing in this simulation.
And that's what we know from the biology, that there's an expanding wave of cell shape changes which is accomplishing the inversion.
But there's a different. But you could say the curvature, because you've got a hole, there is a difference between the curvature and you can have a curvature which is negative and a curvature which is positive. And you could start out by saying that the preferred curvature has the wrong sign for the curvature of the spherical shape that you have.
Correct.
And because you've got a hole, then you could presumably then consider you've now got something which is not in the right shape.
Yes, yes.
And it could invert in that. It could invert in that.
It could, but it appears that there is this. So it could be, yes, it could have been prepared in the wrong state and it's going to the new one. But it appears that it's been prepared kind of in a state which is sort of neutral with respect to that. And then you impose these preferred curvatures through the cell shape change.
But then you've got this. If the spontaneous curvature is localized. That means if you've got a model like that, that means that you have a specific area on the initial surface where there is a spontaneous curvature. Away from it, there is no spontaneous curvature. Initially, right. So now this red region has got to somehow expand. So
Correct.
there has to be some mechanism for the movement of those objects which are defining.
You're getting to the essential biological question, which is what is responsible for this wave of cell shape changes? We know it occurs. We're just encoding it in the theory. The question is, how do you, who've already changed your shape, tell the fellow next to you, okay, now you change your shape, and then you change your shape? What is communicating this information? Is it just elastic effects? There are cytoplasmic bridges. It could be a biochemical discussion. Nobody knows.
All right. Okay.
Yeah, so I absolutely agree with you.
Ray, if you cut a little piece of your red sphere now, will it stay that shape with the two positive curvatures? So
No, I don't think so.
So it might not have spontaneous or two spontaneous curvature in the end, right? It might just have relaxed. It might only have one, for instance.
Yeah.
Quite possible.
Yep.
Could you cut out a piece of tissue in the region where you assume it takes on a prescribed curvature and glue that onto the other side and see whether it...
That might be a little tricky, but the idea of cutting it in some way to see whether there's residual stress has been tried long ago using glass needles of one sort or another, and we definitely want to try to do that.
You've shown how you can suck in a part.
So what I hid from you here is there's a subtle point, which is that these embryos live inside something called an embryonic vesicle, which is a kind of membrane-like structure that holds them. And all of this is happening inside this. And if you try to extract them from that, it doesn't work. So some people have succeeded partially, and we're hoping we can learn to do that. So at the moment, we can't kind of manipulate it very much. We have to just watch. But we'd like to get to the point of manipulating it.
So these guys don't have a cell wall, right? Because if they can change their shape, I mean, they're plant cells, and they don't have a cell wall.
They do, but they change their shape.
But I thought cell walls are fairly rigid, which would mean that the cell wouldn't really be able to
These are single cell, algae, so they don't have a proper...
It's not cellulose, they're hemicellulose, right? It's not a rigid cell wall.
Okay,
No.
it's not rigid, good, okay, great.
Yeah.
But I think that I think you showed that there's something inside which has a particular shape, which is changing the curvature. You had in your picture you had these little ellipsoid type things, and they change as you went round. They were actually changing in shape.
Yes.
And so that, as far as I understood, this is what you would say is where it's initiated.
It's initiated just south of the equator.
Right.
And then spreads.
But if you're saying there is a spontaneous curve, if you want to say there's spontaneous curvature everywhere, which is your red region, right? You might think that the whole exterior of this object is now coated with these objects. I'm a mathematician. I'm not a biologist. Coated with all of these objects. But that's not the case, I guess. These things are not, these shapes inside are not moving. These objects inside are not actually moving around.
No. It's literally, think of it as a shape transformation that's triggered by some unknown process that spreads. It's like infection, you know. You change your shape, then he changes his shape, and that is quasi-statically bending the whole cell sheet.
Yep. So this is the question is, what is it?
Now,
I think to me it's more plausible to believe that there might be nucleation, not of the shape change of the cell necessarily, but of their orientations that we discussed in respect to Misha's question, and that there's a little nucleus where you get something ordered, so you have two spontaneous curvatures in two different directions, and maybe that nucleus then, the phase transition to the nematic or ordered phase where these ellipsoids line up, that could spread.
yeah, one way or another, there's a spreading. That much we know from before our work, that there's a spreading of cell shape changes. The fundamental question is, what's making it spread? My goodness, I'll never finish.
But you have, I mean, there are plenty of other examples, of course, where we know pretty well what's happening, and you always have these mechanisms of apical contraction, actin via myosin activation, and we have plenty of potential molecular mechanisms where that myosin contraction is inductive between cells.
Yeah.
Whether it's externally diffusive or internally diffusive or mechanically.
All of these particular ideas about what's happening map on to the same kind of model, and you're asking the inverse problem, which I can't yet solve, which is I can see the sequence of shapes. We can match it with an elastic theory, but that doesn't tell us what the chemical signal is. It doesn't tell us anything about that.
But my only point is that biologically, your problem is there are too many ways of doing this. In other words, there are dozens of biological mechanisms, any one of which is sufficient to do this process.
Well, let's be careful. We know from the thin sections about cell shape changes. We know from TEM about the change in the cytoplasmic bridge connectivity, and we know how that migrates. So, this is very hardwired to definite experimental observables, and we know from the inversion A mutants, as they're called, where the cytoplasmic bridges don't move, that if you don't move, you don't invert. You get stuck.
I'm saying at a molecular level,
Sure.
there are many possible circuits that can control this, all of which will give the same outcome. Of course, which one or combination is active in your particular organism is always an open question. But there isn't a problem from a biological point of view of coming up with cell mechanisms or regulatory mechanisms that are sufficient to do this. Whether those are the right ones for your organism is a question.
Yeah, and I should say that our view on this was we were testing hypothesis. Most of the time in physics and math, we don't sort of act like we're testing hypothesis. But we're testing hypothesis. The hypothesis is a quasi-static sequence of cell shape changes, which are interpreted as changes in spontaneous curvature, changes in contraction and stretches, can invert the sphere. That's a question. And the answer is yes. Now, that's not a proof that this is the underlying mechanism, but it's a proof of consistency. And that was the first thing we were trying to do. That's all. I'm very modest about this. Yes?
How much variation is there in the location where this spontaneous curvature emerges?
It's actually pretty robust.
How does it affect the time sequence? Is it lengthened if you're up closer to the equator? Or
Well, it's pretty robust, actually. It's always just south of the equator. It's a fairly well-defined region in terms of, let's say, the fraction of the circumference that's occupied initially. And actually, that gets me, if I may, to one or two last things I wanted to say, which is that if you start looking at what would happen if you change the width of this region, then you can start asking about other types of possible behavior. So for instance, suppose you...
One last question.
Okay.
I also see that there is, of course, an asymmetry in the sense that the region where you expand, of course, is toward the South Pole, in that sense.
Yes, exactly.
Yeah, how this is driven?
I don't know. All I can say, I'm sorry, all I can say so many times is I don't know, because we don't have a molecular understanding of how this sequence of cell shape changes propagates. The community, doesn't have it. And that's one of the big goals. But you have to crawl before you can walk, and you have to invert before you can swim. How's that? So we're just trying to see what we can learn and what's the right next question to ask. So here you can do the following. You can ask, as you move around this, let's suppose there's some maximum arc length position where this region of contraction reaches, but the width of it is variable. You know, it could be small, it could be large. And the depth of the preferred curvature that is induced there could itself be another parameter. So you have a sort of two-parameter family of shapes you can go through, and then you can just ask, what is the sequence of shapes as I do that? And you can make a plot of what the curve on which those shapes live as you change the width of the region of preferred curvature. And what you find is this something that looks a lot like van der Waals gas. That is to say, there are parameter values for which if you imagine changing, by the way, I need to define one more thing. If you look at the distance that you move up as you make this transformation, that's what we're plotting here. So we're looking at the depth of the preferred curvature quantity rescaled by r, and we're looking at the distance that you move up as a kind of order parameter for the transition. And you find regions which are perfectly mechanically stable if this length scale is below a critical value. But if this length scale is too large, then you actually get unstable regions. So there's a critical point, and in between, you'd have these huge hops between different possible shapes. The system would be very unstable and out of control. And so it appears, since we don't see that experimentally, it appears that nature has chosen to avoid all of this unstable region and move itself around. But it could have been in a region where you'd get this big instability and coexistence between very different states. Okay, so let me just close.
Pods and plants do that.
Yes, right. There are examples, we know, where there are big mechanical instabilities. So the last thing to say is that I told you in the beginning that in order for this organism to swim, there has to be a large-scale coordination of the orientation of the flagella. Otherwise, they cancel each other out. So they beat from the North Pole to the South Pole with this slight east-west twist. And if you look at the anterior pole of Volvox and you have a good microscope, you can see the eye spots on each of these cells. They point away from the North Pole. They point down and out, and they're highly organized. I mean, if you were evolving for 500 million years, you would be also. And if you take a colony and you smush it against a glass cover slip, and then you draw an arrow from the center of each somatic cell to the eye spot, you have a little spin-like variable that tells you the orientation of the cell in this cell sheet. And as you can see, it's ferromagnetically ordered. It's not perfect. There's a bit of splay. There's about a 10 or 15 degree sort of wander. But there's long-range order, and in other contexts, this kind of thing is called planar cell polarity. And there's a really interesting question of how this arises in the system. Now, it's sort of hardwired by the cytoplasmic bridge connections that occur, but by the time the adult has formed and this extracellular matrix has added, as you see, there are no cytoplasmic bridges anywhere. They've all broken. Now, some adult species maintain the bridges, but most do not. So this order has been maintained as this extracellular matrix has been added and everybody moves apart from everybody else. And I think that's a really interesting question. How does that occur? Is it trivial, or does it require some regulation, et cetera? And in the service of that question, I should say that because we now have this three-dimensional imaging, we can locate every cell on the surface, and we can make a Voronoi tessellation of the surface, and we can start to ask questions about the neighborhood statistics of any individual cell. And this is just literally a few weeks old, but you can actually get the position of every cell on the surface and do what I just said and start asking where are the 5-sided and where are the 7-sided neighborhoods, as there must be if you're going to have a closed object with the topology of a sphere. And is there any pattern from one organism to the next? Do these defects, as you might want to call them, line up in any way? Do they cluster in any way, or is this just totally random? So hopefully the next time I see you, maybe by the end of this workshop, or not the workshop, the longer program, we'll have an answer to that. Okay, so thank you for all these wonderful questions. Let me just remind you that the experiments were done by Stephanie Hohn and Aurelia Hanekamp-Smith, and the theory with me has been with Pierre and Philip. Thanks. Yeah.
Look at the initial stage is and where there's the nucleation of this process. If you look at the curvature, is the curvature different? Is it changing its sign basically? Because this is what we're seeing. You could say that there's initially the curvature has one sign and then it wants to go into another sign. I mean, at the end of the day, because we've got a hole, right? It could be spontaneous curvature could be plus 1 over r or minus 1 over r.
Okay, so we start with something which, morally speaking, is a sphere.
Yes, yeah, but spontaneous curvature might have the opposite. The spontaneous curvature might have a different sign to that.
Of course.
Right.
So, in fact, that's why the graph of spontaneous curvature went down negative and came back, the one that we imposed. So we hypothesized, we transcribed the thin sectioning images. We looked at the width of that region where the cells had shape changes and the cytoplasmic bridges moved. And we say, we know how wide that region is. We know it's tending to splay this way, so we'll make a negative preferred curvature at that location of a certain depth. Okay? Now, I could have said, well, by the time it bends around like this, there's a region of negative curvature, but there's an adjacent region of positive curvature. I could have said, ignoring the thin section data, no, instead I'm going to put a region of positive curvature only there. And that's the smiley face picture I showed you that's wrong. If you do only that, you'll get something that pinches out. And so that's wrong. And that's the lesson, that on a closed surface, you can't trivially read off the preferred curvature from what actually is the equilibrium shape. Now, you could say, well, I should have a preferred curvature of one sign here and another sign there. Yes, of course, I can mix and match. But the most parsimonious description is a negative curvature in the region where we know it is negative and nothing else, and that produces a shape which is consistent with experiment. Does that make sense?
You don't have a closed surface. It's a hole.
Yes, but...
And you could have a closed surface where the spontaneous curvature has got the wrong sign.
I don't know whether
But you could have an equilibrium. You could have an equilibrium, I guess. You could.
So I'm just trying to guess, are you getting to an issue involving the integral of the Gaussian curvature not being constant in this problem because of the
really.
hole? Okay, good. We can remove that.
Well, you could go, you could go into that issue.
It doesn't matter as long as you have uniform property of the splay modulus, actually. You automatically doesn't matter.
Yeah, I mean the hole is there all the time anyway. So.
So this is a nice system as you described because it has only 2 types of cells. But the way you describe this process of inversion makes me think that you substitute this complexity of different types of cells with the complexity of transcriptional heterogeneity that creates all this asymmetry, right? So either...
Well, but even in gastrulation, there are regions where the cell shape changes occur and regions where they don't.
Right.
So you have the same issue there. Why do these cells change their shape and others do not? If you look at the recent work from Wieschaus's group, for instance, on a problem in, I think it's in Drosophila, there's this issue of, okay, there's a gene that's expressed here that's creating a product which is triggering cell shape changes in this region and nowhere else. Fine. So it's a very similar kind of issue of regulation of cell shape changes.
Would this system be still good to address that kind of? Can you chop pieces and do RNA-seq and try to figure out the?
That hasn't been done yet, but I'm hopeful that some invasive technique can get at those changes. Yeah.
Very good. This is very good.
This,
The discussion. So this is very.
So, Ray, one thing.
no lunch for me. Yeah.
One thing that, well, it's supposed to be 12:30.
Yeah. Okay. Wait.
Don't have to. One thing that's critical comparing, and you mentioned it, but you didn't really come back to it now that you're doing the cell tracking, is that my understanding is that you don't have relative cell movement here. That the cells, the topology stays the same.
Correct.
Which is not true in a lot of the other invalidation.
That was the migration/intercalation point I made about it. Yeah.
But it's important to come back and say that you don't have. It's that because that does constrain the models that you could use a lot.
Yes.
Compared to some of the other kinds of invagination problems that you might be dealing with.
Absolutely. Now, I should say, we didn't start out saying we're going to study this problem because it's a good warm-up for gastrulation. We studied because it was beautiful, and we're trying to understand it. And if I try to put it in a larger context, I make this point about gastrulation. But it's interesting in its own right. It's remarkably simple, as an observation, and we're just trying to get at what's the essence of how you do this.
Ray,
Yeah.
it looks like the picture in your movie when it closes, it actually rotates, so it's kicks some fluid out of the Volvox as it rotates when it closes itself back. Right. So my question is, it looks also like you have a bit of snapping effect. You go to a point, you turn it side out, and then you reach a critical point where you can let it go. It will by itself collapse on itself. So you might not think of it as a quasi-static steps process, quasi-static up to a point, and then you have a dynamic effect.
Yes.
And I think the fact that you see it rotating right after is all the energy stored elastically is released and pushed into the fluid that does something else.
So that's an excellent point. Even the speed up at time t=0 that we see, we thought initially, is this one of these snap-through processes that we know from macroscopic mechanical devices? And you have to be very careful about that interpretation because those are mostly inertial, and of course this is a low Reynolds number world where there's no real acceleration. But nevertheless, that was kind of an early question in our mind was, is there a point of no return where it just keeps moving mechanically? And I agree with you, it appears at the very end to be like that, and we're trying to understand at what point you could turn off some of these effects and just let it go. But remember, as soon as it's inverted, those flagella are beating, and so that's why it rotates, but it's also indeed at some point closing up and fluid is flowing in and out of the fialopore. Yeah.
Just one last question, because we have the picture and then continue to okay, go ahead.
Can we understand why it starts being inside out? I mean, is there some functional reason?
This is really interesting.
I don't know.
So my standard answer to most of these questions is we don't know. But one thing to say is that this business of, quote, inverting is present even in the smallest cell sheets that are not closed like a sphere, but just a little kind of cup-like shape in gonium. And the description of this is it's one of those just so stories, but it's something like this, that the first versions of multicellularity probably came from incomplete cytokinesis, the things stuck together. If there was an evolutionary advantage to being like that, then okay, they'll be continued. And perhaps it's just something about the geometry of where the cytoplasmic bridges are relative to this slightly non-spherical shape that produces a little bit of a bend. And that's just an unavoidable consequence of a certain part of cell division. But on the other hand, if you want to swim, it's better to have your flagella as much on the outside of the curvature as possible, so nature figured out a way to fix that. And then it kind of just propagated through that it kept fixing this mistake because there were other reasons why, in terms of the connectivity, it made sense to be like that. So I think evolution is probably full of, or development is full of examples where something something occurs and then is fixed. You know, you see it in your own development. And so that appears to be what's going on here. It's just a kind of inherited mistake that can be fixed, and it's fixed, and it's happy with that.
But Ray, this also, again, if you look in amniotes, that when you get cyst formation in semitogenesis, in tubule development, and so on, the apical surface is on the inside, not the outside. And so almost all situations in which you make a sphere, the initial geometry has the apical surface, and your apical surface has got your flagellum on the inside rather than the outside.
Okay.
Good
That
point.
seems to be, now why that is a good question, but it seems to be a universal.
I hadn't appreciated the universality
The
of that.
basement membrane is on the outside,
Yeah.
and the apical surface is on the inside. And so if you want to be facing the outside world, you have to flip that inside out. It happens in I-cup development, it happens in all of these situations, so it's pretty generic.
So then we have a really deep question we need to answer.
Yeah, well, that's why it's really, sorry, I didn't want to take more of your time, but I think it's really a pretty profound question.
Good.
You want these back?
Yes, please. Could you pass?